Hello and welcome to the presentation of the paper The Origin of Life, What is the Question? By Christophe Malterre, Cyril Jean-Colas and Philippe Nge. My little buddy and I will tell you why the origin of life is one question that is highly researched in astrobiology. Since the first experiment that synthesized organic molecules, which is called the Miller-Urey experiment, scientists started searching for the origin of life from the 1950s on, and this in turn gave rise for research of the origin of life, which has been cited by Fry. Many different papers were released focusing on different approaches. Some look at the origin of life in terms of evolution of the being, like Takeuchi et al., while others look at the chemical evolution like Luisi. One of the big problems today are the financial resources, since origin of life research is a very costly thing, which has been pointed out by Benner. One phenomenon studied in science is the origin of life. Although scientific literature often refers to the question of the origin of life, this question can be understood in many ways. In some contexts, the question can be rephrased as where does life we see on Earth come from? Or in other ways as how actual did life appear on Earth? Or even how is it the case that there presently is life on Earth? But the question can also be interpreted quite differently. Some may argue that the pertinent question that lurks behind the origin of life question is not a how actually question, but a how possibly question, such as how possibly did life appear on Earth? Or how plausibly did life appear on Earth? For others, the scope of possibility may concern not just Earth and its history, but is more general and ahistorical, leading to questions such as how possibly might life appear from non-life anywhere in the universe. Depending if humans are involved in the formation of life or not, the explanandum, which is a phenomenon to be explained, can be understood differently. If humans are involved, the question is how possibly might life in general be made to appear from non-life anywhere in the universe? If no human interaction is found, the question can be formulated as how possibly might life in general spontaneously appear from non-life anywhere in the universe? And as you can see by these questions, the big difference is the spontaneous. And because of this difference depends on the types of approaches that are used. In order to help characterize the different goals pursued by scientists in the origin of life science, Scharf et al. Proposed to distinguish 7 types of life. The first is terrestrial actual life, then we have the extraterrestrial life, the non-standard composition life, the non-standard structure life, plausible life, reinvented life, or in silico abstract life. Furthermore, many works show a need to untie the different origin of life questions, and therefore Malterre et al., who wrote this paper, proposed to do so by focusing not firmly on the different types of approaches to the origin of life question, but on the motivations for the origin of life questions. In order to do so, 3 constraints were imposed. The first constraint, as you can see, is the constraint of historical adequacy. This means it needs to be compatible with current knowledge we have about the early Earth. The second one is the constraint of natural spontaneity. This means the transition from non-living matter to living matter should occur natural and spontaneously. The third one is a constraint of similarity with life on Earth, which is the only life we know, and it's also called life as we know it for this case. Now let's dive into the different types of constraints. The first constraint is the constraint of historical adequacy, which assesses whether they are constrained by an objective of historical adequacy or not. As you can see here, this is the history of Earth, and if the questions are constrained by historical adequacy, they need to follow this structure. Some origin of life questions are set in a historical context, typically the context of Earth, and others formulations reject such historical constraints and position themselves in non-constrained and non-historical contexts. As we will see below, With these historical constraints are relaxed, the origin of life questions allow more general explananda. Multiple interpretations become increasingly speculative. If one puts the origin of life question in a historical context, then they want to explain what happened on Earth 4 billion years ago. In this case, the origin of life question is constrained by historical facts and must be consistent with those. This leads to questions like, how did life appear on Earth 4 billion years ago? Following this, more constraints can be added. First is an additional temporal or sequential constraint, which will define a time frame in which the events need to happen and they need to be ordered sequentially. And one of these constraints could be the inclusion of a chronological marker, as you can see at 3.42 billion years, which is the earliest time for which traces of living cells were found, and another marker at 4.56 billion years, which is the initial time of accretion for Earth. Therefore, any historically constrained origin of life question has to be set between those two markers. Also, constraints that concern specific events can be added. These include physico-chemical constraints of possible parts of primordial proto-living systems, for example, types of prebiotically plausible chemical compounds, and environmental constraints, for example, the atmospheric composition, temperature, and the acidity of the ocean. This second constraint is especially notable in the case of research where it is important to conduct experiments in prebiotic analog conditions such as hydrothermal springs, which can recreate a regulative ideal. The late heavy bombardment, which happened roughly 3.8 billion years ago, has removed the traces of the deep past of the Earth, which leads to many uncertainties about this time, like the importance of the global environmental conditions in comparison to more localized and varied microenvironments, such as hydrothermal vents. This uncertainty also includes the types of prebiotic compounds and systems that existed then. Some push the possibilities of the origin of life back into the very first moments of Earth when the crust started to build and therefore became habitable due to the presence of liquid water and a temporary decreasing atmosphere 4.3 billion years ago. For others, the late heavy bombardment was a reset factor. If one considers supposed traces of life already have been present 3.8 billion years ago or only 3.4 billion years ago after the late heavy bombardment, the window of origin of life on Earth varies between a few million years ago to almost a billion years ago. The earliest traces of life analyzed so far do not reveal the chemical composition of these fossilized living entities, and therefore no molecular-based comparison with the last universal common ancestor, also called LUCA, can be found. This leaves the genealogical path of life in its early days unknown. Because of this, it could be possible that these earlier traces are from an extinct taxa and therefore are not important to today's three domains of life, which are the archaea, bacteria, and eukarya. Despite these uncertainties, certain formulations of the origin of life question assume a concept portrayed by an objective of historical adequacy. On the other hand, other formulations of the origin of life question are unbound by historical questions where the transition from non-living matter to living matter can happen at any point in time in experimentally enhanced temporal windows or in any specific order. One origin of life question that is not historically constrained would be what kind of environment would be favorable for the emergence of life. Relaxing the historical constraints applied to the formation of life on Earth in the past also changes the origin of life question to include the formation of extraterrestrial life. Through this, the origin of life question changes to how might alien life appear on planet T? It is important to note that relaxing the constraints on the origin of life question changes its epistemological nature. The most constrained version of the question concerns an explanandum considered to be true. Lesser constrained versions still concern open possibilities or speculations. If the origin of life question is how life originated on Earth, then the explanandum, there is life on Earth and such life was already present on early Earth some 4 billion years ago, is likely the closest to the truth. It could still be unrelated to life that we find today, as I mentioned before, and is an extinct taxa. Nevertheless, the explananda is to be considered as an established one. Origin of life questions without historical constraints open possibilities to yet established things. For example, no one managed to synthesize novel life forms in the laboratory from chemical compounds or either observe any natural transition from non-living matter to living matter. By relaxing the constraint, the question shifts from explaining a known phenomena, for example the origin life, on primitive Earth to explaining imaginable new phenomena and establishing those phenomena. A second way to untie the origin of life question is to see if they appeal to any natural occurring processes. Suppose the origin of life question is strongly constrained by these processes. In this case, the appearance of life is interpreted as a phenomenon that occurs naturally, without any agential interference and appears spontaneously. Imposing this constraint results in the origin of life question being formulated as which natural processes best explain how living matter spontaneously appears from non-living matter. As you can see, this question is agnostic regarding historical constraints, and it is possible to include any process that might happen on any planet in the solar system or even on exoplanets. Relaxing the constraint of natural spontaneity can be achieved in different ways and at varying degrees. At the most constrained end of the spectrum, experiments can be set up in the field at prebiotic analog sites. On the other hand, by relaxing the natural spontaneity constraint, artificial processes that do not actually mimic natural occurring processes but are reasonably transposable to natural environments can be used. They are naturalizable processes. Such processes include the wet-dry cycles that are transposable to tide pools, as well as solution transfers which mimic stream-connected pools. Strongly relaxing the natural spontaneity constraint allows for artificial processes that require highly complex interventions. When this constraint is relaxed even further, processes considered implausible in nature can be used, but only if we relax the constraints to the max. More accurately, one may consider relaxing the natural spontaneity constraint in two ways. Either by relaxing and mobilizing less natural processes or by permitting more intervention. The simple deed of a chemist putting the essential reactants in exact concentrations in pure water is a slight relaxation of the natural spontaneity constraint. This relaxation can be interpreted as a way to accelerate or increase the probability of natural processes. Additionally, man-made settings in the laboratory evade the varying environmental conditions in nature, and because of this, the yield is bigger than it would be. Further relaxation is achieved by artificially parallelizing many reactions to increase the probability of an outcome. This constraint can be even further relaxed by using a complex setup condition while letting things unfold by themselves, or also intervening at stages of the process. This is implicitly the case when one uses experiments with complex compounds like oligo-DNAs which need human and machine intervention for their synthesis. More extreme cases of intervention include non-terrestrial intervention. This would be the case with not direct panspermia and directed panspermia, where intelligent beings spread living entities on purpose. Similarly to the constraint of the historical adequacy, the most likely to be true explananders are the most constrained ones. The presence of billion-year-old traces, the abiotic synthesis of complex organics, and their abundance of meteorites lead to believe that life spontaneously appeared by natural processes on primitive Earth. Explananda that are less constrained by the natural spontaneity are normally more speculative given the present status of scientific knowledge. This means they are not considered as established facts, but as facts that are yet to be established. And furthermore, suppose the extraordinary chemical complexity of prebiotic Earth. In that case, one might think that it would be easier to create life in a more fine-tuned and simpler laboratory, which is not the case. People may argue that this is because of the high complexity which the prebiotic Earth had, and this is unreachable for humans, but necessary for the origin of life. This constraint of similarity to life as we know it is essential in narrowing down the types of life whose beginnings are at stake because we only know the life on Earth. An origin of life question can be formulated as: which processes best explain how life as we know it appears from non-living matter? To answer this origin of life question, it is necessary to assume that life as we know it is well characterized. It can be characterized by 4 sets of characteristics, namely in terms of chemical composition, the first one, and this means it is based on organic chemistry with water as a solvent. Further, the primary molecular species found in those living organisms are well known and resemble chemistry of life as we know it on Earth. Secondly, the biochemical processes are well characterized and occur in known life forms. For example, electron transport chains as well as the citric acid cycle. On a larger scale, this means that dynamic processes concerning ecological networks and evolutionary patterns common to all known living species have also been well investigated. Thirdly, life as we know it can be characterized by environments in which they can live and thrive. It needs electron donors and electron acceptors, sources of carbon, energy, and liquid water. The need of liquid water further imposes restrictions on the physical parameters like environment and pressure. The fourth characteristic is the necessity of some form of genealogical continuity and a common LUCA. The origin of life question can be relaxed along all 4 aspects. Relaxing the first characteristic of the chemical composition allows the inclusion of hypothetic life forms, such as those that are based on silicon instead of carbon. And it also includes, as you can see here, very hypothetical life forms in the picture. If we constrain it by life as we know it, it wouldn't work obviously, because this is not life as we know it. But if we relax this constraint, this life form would be a possible life form that could exist. And when one relaxes the constraint in terms of metabolism and dynamical processes, the origin of life question could include self-sustained artificial cells devoid of reproduction capabilities. But there we already go in a zone where people may say this is not life anymore. So the similarity to life as we know it is not only for the scientists to find the life, but also for people to accept life and think that life is life. Formulating explanandi constrained by the similarity to life as we know it constraint, for example in genealogical continuity, are more likely to be empirically established when comparing to those that are least constrained. This is because the existence of life as we know it is a proven fact. Those 3 dimensions outlined above form a multidimensional conceptual space that enables one to dissociate significantly different ways of specifying origin of life questions. This can be seen in this graphic here that you have on the slide. Depending on the origin of life questions, this place is different depending on the relaxation of these various constraints. If all constraints are high, life is not different from life as we know it on Earth, which is the space A1 with the origin of life question of how did life as we know it spontaneously appear on Earth. Here all the constraints are high. Contrary to this, you can also relax all of the constraints, which is space D2, which is how can one bring about any life. In this case, the question is about investigating the transition from non-living matter to living matter independently of any historical prebiotic plausibility or compliance with life as we know it. Also, there is no constraint about naturally occurring processes, and this is primarily the case in synthetic biology or system chemistry. In between those 2 extremes, any combination of constraints is possible. For instance, one may research an origin of life question with low historical constraints, high natural spontaneity, and low constraints regarding similarity to life as we know it. This would be region B2 on figure 1, with the origin of life question of how does any life spontaneously appear. This is normally the region where astrobiology research falls into. From explanation-seeking questions to fact-establishing questions. In many cases, the phenomena forming the target of the question have not yet been established. Therefore, the origin of life question is rarely an explanation-seeking one. For many of these cases, the phenomena of how did life as we know it spontaneously appear from non-living matter on prebiotic Earth can be reformulated as why P? Or how is it the case that P? While P is the question life as we know it spontaneously appear from non-living matter on prebiotic Earth. If the constraints are still loosely defined, P is true. And as a result, one may request an explanation of the phenomenon described by P. But B could also be false, since currently available evidence is still in line with different hypotheses like direct panspermia or the appearance of life as we know it during a second origin among the remains of a more primordial life. Nevertheless, P is largely considered to be true within the wider scientific community. And when the three constraints are relaxed, the question can no longer be in the form of how is it the case that P, because P has not yet been established and is still hypothetical. Consequently, the question changes to something like how would it be the case that P, if P were true? And very important in the research is that P can either be established or not, in the form of is it the case that P, as can be seen in B2, which we talked about before. For B2, only the natural spontaneity constraint is high, while historical adequacy and similarity to life as we know it is low. P remains a hypothesis for all regions of the conceptual space except for the most constrained ones, which here would be A1, as we said. There, the explanandum is taken as true, because we know that life as we know it today appeared on Earth spontaneously. For other regions, the existence of the phenomena needs to be established, which in turn leads to new instances of the origin of life. Now, the origin of life generally refers to the origin of life without additional specifications, but researchers have to relax constraints because of their usual science practices like methodological constraints or limitations. Relaxing some constraints enables more tractable approaches that are more likely to yield an explanation. It is argued that the value that a given community may help to particular finding, addressing relaxed origin of life questions seems almost necessary to construct a path to addressing the most constrained questions. And furthermore, to study origin of life questions needs intricate combinations of sociological and technical factors, and those yield good results. For example, the study of chirality, which is a very important field of study by itself, have removed burden from the most constrained origin of life questions. Furthermore, the search for life in our solar system or on exoplanet is unavoidable to relax the similarity to life as we know it constraint, since it is very likely that life that is found on exoplanets does not have similarities with life on Earth. Now more personal thoughts to this paper. There's like a future outlook that new discoveries might emerge when constraints are relaxed, and so we can find more life the more relaxed the constraints are. And when we find this life, it might also give rise to new scientific fields, which in turn will allow us to find more extraterrestrial life which we were not able to find before. So this means it will build up on itself, and the more we find, the more we will be able to find. Which can you see here? For me, this paper I think was very good, but it was also written in a very complicated English, and I had to read it multiple times to entangle all of their ideas. Furthermore, life could also exist outside of these constraints. Yes, if one relaxes the constraints to their maximum, basically nothing is constrained anymore, but that life might live in different constraints and that they're not even included in those constraints. And also, these constraints might include life that we cannot even identify. So these constraints do not do justice to the life that lives out there, and therefore this study cannot even be used to help identify that life. My little buddy and me say goodbye now, and I hope you enjoyed our presentation, and here you can see the sources that I used for this. Thank you very much for listening.